Welcome back to this next video in which we are discussing the thyroid gland. Okay, so we're currently in the process of discussing the effects of the thyroid hormones T3 and T4 on the peripheral tissues of the body. And remember, I know I keep saying this, but it's worth repeating, T3 and T4 levels do not fluctuate in the bloodstream, so whatever T3 and T4 are doing around the body, they're doing it continuously. Okay, now remember, I've said that we're going to divide the effects of the thyroid hormones on the peripheral tissues of the body into three main categories. The first category we've already seen, and that is that in the background to glucagon and insulin, thyroid hormones are continuously going to be activating the mobilization of energy sources into the bloodstream. So they're mobilizing glucose from hepatocytes into the bloodstream, amino acids from skeletal muscle cells into the bloodstream, and free fatty acids from white adipocytes into the bloodstream. In addition, category number 2, they're going to be continuously activating heat production within the human body. And in the adult human, the way that they do this is by acting on kidney cells and liver cells and increasing the density of leaky sodium and potassium channels in the cell membrane of those cells, and also increasing the concentration or density of sodium potassium ATPases in the cell membrane, and therefore increasing the amount of sodium and potassium that is continuously in flux across that cell membrane, increasing the cycling of sodium and potassium across the cell membrane, and the energy that's used to do that will end up as heat. Now what I want to talk about is another pathway by which T3 and T4 can activate the production of heat continuously in humans. However, this is only believed to be relevant to human neonates, to babies. It's not believed to be relevant for human adults. Okay, so this next pathway is going to be just for human neonates, and it's going to involve special cells known as brown adipocytes or brown adipose tissue, often abbreviated down to BAT. Okay, so this stands for brown adipose tissue. Now, brown adipose tissue, it's present in human neonates, but it's not present in human adults. At least we don't believe it's present in human adults. It is present in the adults of other species of mammals, but we don't think it's present in human adults to any notable level. So we believe that brown adipose tissue and the physiology of brown adipose tissue and the involvement of T3 and T4 in the physiology of brown adipose tissue is just relevant to human neonates. So what is brown adipose tissue? So brown adipose tissue, they're cells. Brown adipocytes are cells, but they have very different function to white adipocytes. They also store triacylglycerols, just like white adipocytes. But white adipocytes, their role is simply to store the triacylglycerols. And then during the fasting state, they break them down and release the free fatty acids into the bloodstream. The brown adipocytes, they have absolutely no intention of releasing these triacylglycerols back into the bloodstream. Instead, what they are going to do is they're going to use them, they're going to catabolize them, and they're going to release the energy from them and as heat. So they're going to use the triacylglycerols to generate heat rather than just to store them. So let me draw you a picture then of a brown adipose tissue cell. So here it is, and it does look slightly different to a white adipose tissue cell. So firstly, the major difference between the two is that a white adipose tissue cell just has a single huge lipid droplet to store all of its triacylglycerols in, whereas a brown adipocyte will have multiple lipid droplets. So in its cytoplasm here, it will have a huge number of lipid droplets like so. So lots of lipid droplets rather than just one massive lipid droplet, as was the case for the white adipose tissue cells. Okay, so here is my picture then of a brown adipocyte. In addition, brown adipocytes have a lot of mitochondria, so I'll just draw these as little sort of structures here. I won't attempt to draw them in any detail, little black blobs here. Okay, so lots of mitochondria, and it's these mitochondria that are going to be the site of heat generation. So what's going to happen is the brown adipocytes are going to use the fatty acids of the triacylglycerols in beta oxidation, which will produce acetyl coenzyme A, and the acetyl coenzyme A will then go through the Krebs cycle. The Krebs cycle will then result in the production of reduced electron carriers, so reduced NAD and reduced FAD, and these will give those electrons to the electron transport chain, which is a bunch of proteins in the inner membrane of the mitochondria, which will use the energy of the electrons to pump protons from the matrix of the mitochondria into the intermembrane space, and then usually, of course, that proton gradient is then used to generate ATP, but in the case of the brown adipose tissue cells, we're going to have what's known as an uncoupling protein, which is going to allow the protons to go back from the intermembrane space to the matrix without actually generating ATP, and the energy released will just go into heat. So let me talk about this in more detail now. So I'll draw a picture then of a mitochondrion in much more detail here, because this is going to be the key site. So remember, mitochondria have two membranes. We believe that they are potentially originated from bacterial cells that were phagocytosed by the precursor to a eukaryotic cell. So the inner membrane is effectively the bacterial cell membrane that got phagocytosed, and the outer membrane is the membrane of the phagosome. Okay, so I'll label up the key portions here. So this is the inner mitochondrial membrane with all of these projections into the central matrix. So this is the inner membrane. Then we've got the matrix of the mitochondrion, which is the space within the center, so the space within the inner membrane. And then these projections are to increase the surface area of the inner membrane of the mitochondria, and they project into the matrix here, and they are known as cristae. Okay, so I'll just underline all of this in pink. So we've got the matrix, the cristae, the inner membrane of the mitochondria. The other key things to label up here are we have the outer membrane of the mitochondrion. So this is the outer membrane, and then very importantly we have a space in between the inner membrane and the outer membrane, and this is known as the intermembrane space, and this is the space where the protons are going to be stored. So this is the intermembrane space. Right, okay, so to once again summarize the metabolic processes that are going to occur. So remember, lots of different metabolic processes converge on the Krebs cycle. So glycolysis and the link reaction for glucose, they produce acetyl coenzyme A, which can enter the Krebs cycle. Beta oxidation for fatty acids also produces acetyl coenzyme A, which converges on the Krebs cycle. The Krebs cycle will occur in the matrix of the mitochondria, and it will generate these reduced electron carriers, so reduced NAD and reduced FAD. The reduced NAD and reduced FAD, they carry electrons, and they then give the electrons to proteins that are in the inner mitochondrial membrane here. So I'll just draw a few of them here, like so, which are known as the proteins of the electron transport chain, so the ETC. And remember, these proteins are massive great complexes of loads of separate polypeptides, and they pass the electrons from one to the other, and they gradually extract energy from those electrons, and they use this energy to move protons from the lumen of the the matrix of the mitochondria to the intermembrane space. Okay, so overall this is where the energy of all of these catabolic reactions is ending up being stored, in this establishing proton gradient across the inner membrane of the mitochondrion. So we end up moving loads of protons from the matrix of the mitochondria into the intermembrane space. Now this creates both a concentration gradient of protons but also an electrical gradient, because you're moving positive charge from the lumen into the intermembrane space, and you actually end up with a massive great electrical potential difference across the inner membrane of the mitochondria of negative 160 millivolts. So the electrical potential inside the matrix is 160 millivolts lower than the electrical potential in the intermembrane space, because you've moved all of this positive charge into the intermembrane space. So that's the reason the protons don't just diffuse off, because they're being pulled back against the inner membrane of the mitochondrion by this very large electrical potential difference across the inner membrane. Okay, so you end up with lots of protons stored in the intermembrane space then. Now, what can then happen is there is a protein in the inner membrane of the mitochondria to allow protons to move back from the intermembrane space into the matrix of the mitochondria, and then it will use the energy released by this movement of the protons to synthesize ATP, and this is of course the ATP synthase protein. Okay, so this can allow protons to move back from the intermembrane space into the matrix, and it will use the energy to synthesize ATP. However, in brown adipose tissue cells, we're going to have another protein in the inner membrane of the mitochondria, which will also allow protons to move back from the intermembrane space to the matrix of the mitochondrion, but will not use the energy released by that movement of the proton down its electrochemical gradient to synthesize ATP. Instead, the energy will just go into heat. And this protein is known as the uncoupling protein 1. So here it is, it's called UCP1, the uncoupling protein 1. So I'll colour it in green here, and I suppose I should write out its name in full. So UCP1 stands for the uncoupling, the UC is for uncoupling, and then the P is for protein, and it's the uncoupling protein 1. Right, so the basic principle of this is it's just going to allow protons to move from the intermembrane space back into the matrix of the mitochondrion, and it won't harvest the energy that's released when the proton moves down its electrochemical gradient to do anything. It won't use it to synthesize ATP. Instead, it just goes into heat, and that's the means by which brown adipocytes actually produce heat. Okay, by this uncoupling protein 1. So that's the mechanism by which brown adipocytes actually take part in thermogenesis then. So what does T3 and T4 have to do with this? Well, T3 and T4, they continuously drive the brown adipocytes to produce uncoupling protein 1. So they drive the synthesis of the uncoupling protein 1. They also drive the synthesis of another important protein, which is a receptor that the brown adipocytes can put on their cell membrane, which is the Betafree adrenerreceptor. A rare adrenerreceptor that you don't hear much about, but which is important, particularly on adipocytes. Okay, so what then is the role of the Betafree adrenerreceptor? Well, one of the things that drives brown adipocytes to actually participate in their function, of course, is T3 and T4, but also adrenergic stimulation, both by noradrenaline and adrenaline, also drives brown adipocytes to participate in thermogenesis, and this is through the beta-3 adrenergic receptor. So by elevating the beta-3 adrenergic receptor on the surface of brown adipocytes, T3 and T4 are actually going to increase the amount of thermogenesis occurring inside the brown adipocytes. So let me just go over the means by which the beta-3 adrenergic receptor actually activates thermogenesis within the brown adipocytes. So the activation of the beta-3 adrenergic receptor is going to stimulate more beta oxidation to actually occur, more breakdown of free fatty acids, well, more breakdown of triacylglycerols that are stored in these lipid droplets to release free fatty acids, which can then be used in beta oxidation to drive the formation of a proton gradient across the inner membrane of the mitochondria inside this brown adipocyte. So if I actually draw this process on, so let's just have this representing the cell membrane of our adipocyte here. So we'll have beta-3 adrenergic receptors in the cell membrane of the adipocyte, like so. So this is the beta-3 adrenergic receptor, and this can be stimulated then by either noradrenaline or adrenaline, which I'll just refer to as NA/A here. And of course, when the beta-3 adrenergic receptor is activated, this is going to activate the GS heterotrimeric G protein. So all of the beta adrenoreceptors, beta-1, beta-2, beta-3, they all act through the GS heterotrimeric G protein. So I'll draw this here. So of course, the GS heterotrimeric G protein, it's a heterotrimer of 3 separate proteins. the alpha protein, which is specifically the alpha S protein, the beta protein, and the gamma protein. Okay, and the beta and gamma, there's loads of different types of beta and gamma proteins that can make up heterotrimeric G proteins, but for this to be a GS heterotrimeric G protein, that doesn't specify what the beta and the gamma have to be. All it means is that the alpha subunit has to be an alpha S subunit. Okay, so this is the important one. It doesn't matter what these two are. Okay, so we'll just call them collectively beta and gamma. Right. So, GS heterotrimeric G proteins, they have two states. They have an on state and an off state. Now initially it's going to be in the off state, and in the off state the alpha subunit has a GDP molecule, guanosine diphosphate molecule, bound to it. And when the alpha S subunit has a GDP molecule bound to it, it likes to bind to the beta and gamma subunits. Now the beta and gamma subunits, they always remain together. They're always bound together, and they are often referred to collectively as the beta-gamma complex. Whereas, once this has actually been activated and we've replaced the GDP with a GTP, the alpha S is no longer going to want to bind to the beta-gamma complex, so they're going to separate. But at the moment, it's in the off state, this GS heterotrimeric G protein, so the alpha S subunit has GDP bound to it, and it, in this state, wants to bind to the beta-gamma complex. Now the whole thing is attached to the underside of the cell membrane. Now, so this is in the off state. We've got GS heterotrimeric G proteins floating around, attached to the underside of the cell membrane, not doing anything. We've got the Betafree adrenergic receptor, which has not yet been stimulated. Now when the Betafree adrenergic receptor is stimulated by noradrenaline or adrenaline, this receptor is going to change conformation. Specifically, the intracellular portion is going to change conformation, and now it actually forms a docking site that can bind to the GS heterotrimeric G protein. So this can come along and bind to the underside of this beta-free adrenaline receptor, and binding here will catalyze the activation of the GS heterotrimeric G protein. So let's say this is now bound to the underside here. What it catalyzes is the release of the guanosine diphosphate from the alpha S subunit, and then the binding of a guanosine triphosphate molecule from the cytoplasm of the cell into that site where the guanosine diphosphate was. The instant the GTP binds to the alpha S subunit instead of the GDP, the alpha S subunit changes conformation, and this causes it to cleave away from the beta-gamma complex, and everything also cleaves away from the activated beta-free adrenergic receptor. Okay, so it was for a while bound to the underside of the beta-free adrenergic receptor. Once you've had the activation of the GS heterotrimeric G protein, however, everything moves away. So here now is our alpha S subunit. So this is a progression. So this is picture number 1. This is now picture number 2. Okay, so here is the same cell membrane. Here now is the alpha S subunit, again in green here, which now has guanosine triphosphate bound to it, like so, and then the beta-gamma complex will now be separate from it, and it also has signalling roles, but they're not going to be ghastly important here. We're going to mainly be focusing on, well, completely focusing on the role of the alpha S subunit in signalling. Okay, so we've got the activation of the heterotrimeric G protein now. The GS heterotrimeric G protein is in its activated state. We have the alpha S subunit bound to GTP, and it's now segregated off from the beta-gamma complex. This one is the one we're going to follow. It's going to now go and activate an enzyme that is sitting in the cell membrane over here by the name of adenylyl cyclase. Okay, so for short we can abbreviate adenylyl cyclase down to AC here. And I'll just colour adenylyl cyclase in. So this is a membrane-bound enzyme, and it's going to be activated by the GS heterotrimeric G protein once it's in the activated state, specifically the alpha S subunit of the GS heterotrimeric G protein. So the alpha S GTP subunit comes along, binds to the adenylyl cyclase, activates it, and now the adenylyl cyclase is going to start catalyzing the reaction which it is famous for. And the reaction which it catalyzes is the conversion of ATP, adenosine triphosphate, into cyclic AMP, CAMP, plus a pyrophosphate molecule. So we get the production of cyclic AMP then in the brown adipocytes upon the stimulation of these beta-free adrenergic receptors. Cyclic AMP will then go off and activate an enzyme in the cytoplasm of the cell known as protein kinase A, or pKa for short. I'm sure you've heard of these enzymes before, they're very, very famous. Okay, so here is protein kinase A, like so, in the cytoplasm of the cell. And protein kinase A, what's it now going to do? It's a kinase enzyme, and it phosphorylates other proteins, so it can stick phosphate groups onto the serine and threonine residues of other proteins, and by adding these phosphate groups onto other proteins it can change their function. So specifically in this case, in these brown adipocytes, it is going to phosphorylate and activate an enzyme known as hormone-sensitive lipase, HSL. Okay, so this is hormone-sensitive lipase because it responds, its activity inside the adipocytes is determined by hormones such as adrenaline. Okay, so hormone-sensitive lipase. So protein kinase A will phosphorylate and activate the hormone-sensitive lipase enzyme, and this enzyme now goes to the lipid droplets inside the brown adipocyte and is going to start breaking down triacylglycerols to release free fatty acids. Those free fatty acids are then going to be catabolized by beta oxidation to produce acetyl coenzyme A, and you're therefore going to get more of the Krebs cycle occurring, and therefore more of protons being moved from the matrix of the mitochondria into the intermembrane space, and therefore more protons being moved back into the matrix by the uncoupling protein 1, and therefore more heat production. So T3 and T4, they are involved in making sure that there's a high expression of the uncoupling protein 1, and they're involved in making sure there's a high concentration of these beta-free adrenoreceptors on the surface of the brown adipocytes, but it's noradrenaline and adrenaline that actually stimulate the breakdown of triacylglycerols into free fatty acids so that the metabolic processes can actually be upregulated. So adrenaline and noradrenaline are the ones that actually stimulate the brown adipocytes to generate heat. T3 and T4, if you like, they're just making sure that the necessary apparatus is there. For instance, the uncoupling protein 1 needs to be there. They also increase the sensitivity of the brown adipocytes to adrenaline and noradrenaline by increasing the expression of the beta-free adrenoreceptors on the surface of the brown adipocytes. So overall, T3 and T4 are very important in human neonates in maintaining the normal physiology of brown adipocytes and therefore maintaining normal thermogenesis. Okay, right, so now moving away from category 2 of heat production, let's go on to the final category that I'm going to talk about of the effects of T3 and T4, and this is the effect of them on the heart. So T3 and T4 then, what is the effect that they're going to have on the heart? Well, they're going to go into cardiomyocytes all over the heart, and they drive increased expression of a receptor on the surface of the cardiomyocytes. Specifically, they drive the expression of the beta-1 adrenoreceptor, so another adrenoreceptor, indeed another beta adrenoreceptor. So they're going to increase the expression on cardiomyocytes of the beta-1 adrenoreceptor. So let me just draw a little picture of this. So if we have our cardiomyocyte here, the beta-1 adrenoreceptor is another G protein coupled receptor. So here is my classic picture for G protein coupled receptor. So they're going to increase the expression of beta-1 adrenoreceptors on cardiomyocytes all over the heart. Now, what does that mean? They are involved in making sure that sympathetic tone to the heart is normal. Now, again, I'll remind you that T3 and T4 are there continuously, their level doesn't fluctuate, so they're doing this all the time, so they're always driving the expression of beta-1 adrenoreceptors on the heart, and if you had too much T3 or T4 because of some pathological reason, then you'd end up with too high beta-1 adrenoreceptor concentration on the cardiomyocyte cell membranes, too little T3 and T4, and you'd end up with too little beta-1 adrenoreceptors on the surface of the cardiomyocyte. So remember, they are doing this all the time, and fluctuations are going to lead to abnormalities in the physiology all around the body. Okay, so they maintain a normal level of sympathetic stimulation onto the cardiomyocytes. So remember, all the time the heart is getting noradrenaline being released onto it by the cardiac nerves, a little bit of noradrenaline, and there might be a little bit of adrenaline in the bloodstream as well, which will also be stimulating cardiomyocytes. T3 and T4 are involved in making sure that the cardiomyocytes have their normal receptivity to adrenaline and noradrenaline by making sure that they have the normal level of beta-1 adrenoreceptors on their cell membranes. Now, what does sympathetic stimulation then do to the heart? And by the way, this normal level of sympathetic stimulation onto the cardiomyocytes is then called SNS tone. So that's what this means. Tone means a tonic level of stimulation. The sympathetic nervous system, through the cardiac nerves releasing noradrenaline onto the cardiomyocytes, is going to be stimulating the cardiomyocytes continuously, provided they have the normal level of receptivity, i.e. the normal level of receptors on their cell membranes. This continuous sympathetic nervous system stimulation of the cardiomyocytes is referred to as SNS tone. Okay, and what's it responsible for doing? Well, sympathetic nervous system stimulation of cardiomyocytes, it has two major effects. In the cardiomyocytes of the sinoatrial node, it increases the rate at which those cardiomyocytes generate electrical signals, and therefore, because those electrical signals drive the entire heart rate, it increases heart rate. In addition, on normal contractile cardiomyocytes, sympathetic nervous system stimulation increases the contractility of the heart. So these are the two major effects then of the sympathetic nervous system on the heart, increasing the contractility of the cardiomyocytes, which means how hard the cardiomyocytes are actually going to contract when they receive an electrical signal to do so, how much force they're going to generate when they contract, and also increasing the heart rate, increasing how often the cardiomyocytes are going to contract. Okay, so there is a normal amount of sympathetic stimulation on the heart. The sympathetic nervous system always is stimulating the heart, and that maintains a normal heart rate. Okay, so if you were to lose that sympathetic stimulation, then the heart rate would go down and the contractility would go down. So there's always a little bit of sympathetic stimulation on the heart, known as sympathetic tone, which is maintaining heart rate nice and high at a normal rate of around 75 beats per minute, and also maintaining normal cardiac contractility. And if you were to lose that, then you'd get a lower heart rate than normal, bradycardia, and you'd end up with too low contractility of the cardiomyocytes. Okay, so T3 and T4 then are involved in regulating this normal level of sympathetic nervous system tone on the heart by making sure that the cardiomyocytes express the correct and normal level of beta-1 adrenoreceptors, which is the receptor by which noradrenaline and adrenaline act on cardiomyocytes. Okay, so to emphasize this, if you had too high levels of T3 and T4, you'd end up with far too high beta-1 adrenaline receptor expression on the cardiomyocytes. You'd therefore end up with far too high heart rate and far too high contractility, because the amount of noradrenaline and adrenaline that you're releasing onto the heart hasn't changed, but now the responsiveness of the cardiomyocytes has changed to that. So you're going to get more stimulation of the cardiomyocytes. If you had too low T3 and T4, then you'd have too low expression of beta-1 adrenoreceptors on the surface of the cardiomyocytes. This would lead to too low responsiveness to the adrenaline and the noradrenaline that is stimulating the cardiomyocytes, and therefore you'd end up with too low heart rate and too low contractility. So T3 and T4 is involved in continuously maintaining normal responsiveness of cardiomyocytes to sympathetic nervous system molecules such as noradrenaline and adrenaline. Okay, right, so that now concludes our discussion then of the peripheral effects of the thyroid hormones T3 and T4. In the next video, what we'll turn our attention onto is the control of the release of the thyroid hormones by the thyroid gland, which is done by the hypothalamic-Pituitary-Thyroid axis.